if we can make materials that act like living cells? That's exactly what the researchers in the Brandeis MRSEC are doing. I'm Robert Keane, a graduate student, and I'm going to take you on a tour and show you how we work together to understand the rules of life. In our center, theorists and experimentalists closely collaborate in this quest. We have our own computer cluster, which, along with external resources, we use to carry out simulations relevant to self-assembly and active matter. Now I want you to meet some of the scientists in the Brandeis MRSEC who seek to understand and mimic the beautiful phenomena we see every day in the living world around us. Anjali is a postdoc in the Rogers Lab who works on self-assembly. One of the grand goals of the Brandeis MRSEC is to assemble synthetic materials with specific sizes, shapes, and functionalities, much like how biology creates small things like proteins, all the way up to entire organisms. So how do we do that? Well, one way is with the geometry of the individual building blocks. Take, for example, this pentagonal block. It has faces that are designed to attach at specific angles with its neighbors. When you mix any of them together and give them a little shake, kind of like what happens inside of a cell, they assemble, in this case, into something that might resemble the shell of a virus. One that you might be familiar with is this one. Or maybe you instead want to assemble tube-like structures with different lengths or membranes with some specific width and height. These building blocks are like the pieces of a puzzle. By tuning how they fit together to their shapes and also their properties, in this case, simply their colors, we can assemble individual pieces into whatever larger object we want. Now, while all these things we can 3D print and hold in our hands, we've begun to realize these assemblies on the micron scale, something like a tenth or even 100th the width of a single strand of hair. This is just the beginning of the quest to design puzzle pieces of our synthetic materials. And in the future, we can create larger structures with specific sizes and shapes by simply tuning the geometry and the interaction of these smaller pieces. Here we are at the Brandeis Athletic Center, on our way to see Salman, another graduate student in the MRSEC here at Brandeis. Let's see what he's up to today. Salman studies movement at the macroscopic scale that's generated by millions of tiny proteins in cells, synergistically dancing around to the beat of life. Now let's head to where Salman works. On the way, let me describe a little bit more about what he does. Cells are the fundamental building blocks of all life. They are capable of performing complex tasks such as self-replication and movement that lets them solve mazes. These tasks result from a coordinated effort by components of the cell, such as the cytoskeleton, molecular motors, and ATP, the fuel of life. At Brandeis, scientists extract these components and characterize them through reconstituted, well-designed experimental setups in order to understand emergent behavior. Using fundamental building blocks like microtubules, which are rod-like filaments that form the backbone of the cytoskeleton, and kinesin motor proteins that are the walkers on the microtubule mesh, scientists can create systems that consume energy at the microscale to generate large-scale, lifelike collective behavior. Advanced experimental techniques like microfluidics, microscopy, and theoretical techniques like machine learning and numerical simulations are the tools that invent protocols to control macroscopic properties at these lifelike systems. These efforts have led them to create systems that can live for days, materials that can be controlled by shining light on them, and even materials that behave both like solid or liquid on demand. Behavior that is not expected in conventional materials. With the advancement in technology and an intermixing of expertise from biology, physics, chemistry, mathematics, and computer science here at Brandeis MRSEC, scientists hope to measure and characterize movement and forces at the smallest of scales. This understanding will help us to design and control lifelike systems that leverage principles of life to perform complex tasks which are not possible by otherwise dead systems. The orchestra of careful design, intelligent measurement, and creative control is the song of life that scientists here at Brandeis sing to create futuristic lifelike materials. Thanks for joining me today. I hope you enjoyed the Brandeis MRSEC with me.